vessels, and even uniformly beating heart cells in a petri dish. To confirm that all of these different types of cells could be produced from one red cell, instead of many red cells that can each make one type of tissue, they took single cells, allowed them to divide, and then did the same assays looking for their ability to produce breast, pancreatic, fat, blood vessel, and heart cells. And the scientists found that a single red cell could produce all of these tissue cells. So all of this data shows that these red cells, isolated from normal breast tissue from multiple women, have the ability to produce all kinds of different cells that are perfectly functional. So next, the scientists were curious to see if these red cells, like embryonic stem cells, could divide forever, which could suggest that they may be on their way to becoming cancer cells and could make it more difficult to use these cells for therapy. But surprisingly, the scientists found that these red cells could not divide forever. They had a very limited lifespan, unlike embryonic stem cells and many cancer cells as well. They also had perfectly normal chromosomes or DNA profiles. So these two aspects of their biology suggest that they are probably not precancerous cells that tend to have a lot of changes at the level of the DNA. So what did this study show? This study found two markers that could differentiate a cell that has the ability to make different tissues from a cell that does not: high CD73 and low CD90. The scientists call these cells endogenous pluripotent